is quite important. So that means that seaweed is a commodity that in the market fetches a low price. And then on the contrary, the crustaceans, this is nearly 30% in value. We also know that this is particularly because of the shrimp production. Crab, lobster, prawn are coming up, but not at the speed that we have seen with shrimp. Maybe not for you, but for us in the Western world, this is a big surprise, because for us in the Western world, mollusks, when you think about oysters, when you think about scallop, when you think about mussels, these are high value. This is, I would say, luxury food. Well, nonetheless, in many countries, and particularly in Asia, value of mollusks is low, which means that that high quality animal protein is available at a very reasonable price. I put in this slide because for me it was the first time that I have seen FAO presenting the data, the comparison between fisheries and aquaculture. And it's interesting to see that indeed, yeah, in the red here, aquaculture, this is a very important contribution already. And for some, when we talk about the inland fish and we talk about seaweed, we see that the majority, the far majority, is coming from aquaculture. And that it is, when we talk about marine fish, that it is the opposite. The majority comes from fisheries, and in fact only a small fraction comes from aquaculture. And this is where in some regions in the world there is a misconception, like in Europe. In Europe we think aquaculture is only salmon and sea bass and sea bream. No, this is okay the case in Europe, but we should not extrapolate to the rest of the world. Okay, so that was a very, very brief, let's say, review on data and the recent evolution. But in the same document, FAO 2022, we see what challenges we are facing for the years to come. And these challenges have mainly to do with availability of food. And I'm not just mentioning aquatic food, I'm talking food in general. And we know the population is still increasing. We see that the buying power, so that the poverty is decreasing, and in particularly Asian countries. I'm not so familiar with India, so I cannot speak for India, but I know that China, for example, as soon as the people have some better financial means, one of the first things they invest in is food, and very often even seafood. So FAO has made a calculation, FAO has checked what is available from fisheries, what should then be filled in by aquaculture. And I must say, I'm not so optimistic as FAO that in order to meet the global demand by 2030, which is only a little bit more than a half of a decade away from us, we will have to produce 30 to 40% more aquatic products per year. And I think this is a big, big challenge, particularly when you know that there are quite a number of issues at stake. We do not have the time to go through all the topics that are listed here. I will mainly zoom in on environmental impact and on diseases, but there are many, many other issues that really provide a threat for the further developments. And the one in Europe, for example, I don't know again for India, but in Europe we have such, not only stringent, because I'm the first one to say we should be very strict when it comes to issuing permits, when we have to allow new developments in aquaculture. We have to think about the environment. But the problem is that in our Western world, we have so many different entities, Ministry of the Environment, Ministry of Agriculture and Fisheries, Ministry of Industrial Activities, and these are like ivory towers. And when you need a permit for a new farm, well, in the average in Europe is minimum 2 years. And that's very, very sad, because a lot of young people that might be interested and might be motivated to go into aquaculture, well, they don't have the financial means to wait another 2 years to be able to start, or they look for other opportunities. So this is one of the reasons that in the past 5 years, aquaculture is stagnating in Europe. And Europe is very, very much concerned, just to mention you that there are quite a number of issues at stake. Before I proceed, particularly on the two items that I was mentioning in the previous slide, environmental issues and diseases, let me go back a bit in history, and you will see that I have a good reason to go back and compare. You will not find this definition in textbooks. That's one I came up with. And I'm classifying aquaculture in two big, let's say, historical and still activities today. And I call one the food aquaculture, and the other one in a moment you will see I call the business aquaculture. Food aquaculture is the type of aquaculture where you produce food for local consumption. Yeah, and this is an aquaculture that has a very long history, more than 2,000 years, particularly in China, but I'm sure that also in your country you have records of aquaculture practices, probably also in the millennia back. It's a type of aquaculture that is family-based. It's most of the time people involved in production of be it carp or be it some other mostly fish for local consumption. So that's why I call it food aquaculture. It's producing food for local consumption. Low technology, well, also the fact that there was limited expertise, limited knowledge on the biology. In many cases it is quite labor intensive, but crucial. And you remember my discussion at the start and the title. This food aquaculture with such a long history is one that is based on integrated farming. And I don't have to give you examples here from Indonesia. I'm sure you are still well familiar with cases in India, where you have either goats or chicken, different integrated aquaculture practices with terrestrial animal breeding. And of course, the key example, which sad enough disappeared at a certain point of time, the integrated rice plus fish and crustacean production, disappeared at the moment that people wanted to see higher yields with the rice, when the time came that they were using herbicides, pesticides. But the good news is that I would say the last 5 years, maybe a bit more already than 5 years now, organic rice farming is becoming popular. The country that is most successful with organic rice farming is China. Already 2 million hectares under organic practices, and which means that since no more chemicals are used, one can go back to the old practice of integrating with aquaculture. And it is there that in fact all aquaculture started. I'm sure in some of your classes on aquaculture you heard that the first textbook, can you imagine, the first textbook on aquaculture was published 2,490 years ago, Van Lee, the integrated farming of rice, the rice paddies, and the carp. So this is where it all started, and this is, let's say, to finish here on food aquaculture, this is the one that has the longest history, and happy enough to see that it is still practiced. I remember the time, I would say some 15 years ago, that my Chinese friends were saying, it's time that we forget about food aquaculture and that we move to business aquaculture. It's very, very good that maybe some practices have changed, but that food aquaculture, as we will hear in a moment, still has a role to play. So what do I mean with business aquaculture? Well, this is, again, of course, production of aquatic products, also as food, but not for local consumption. Business aquaculture is producing aquatic products to do a business, to sell, to make money, and of course use the money maybe to buy food, but maybe also to buy shoes or invest in a better house, you name it. So this is why I'm using the word business aquaculture. And well, the younger people among you will not agree that I still call this a recent development, but okay, when we look into the history of aquaculture, it is only since the 1960s that, in fact, the approach of business aquaculture was initiated. The Japanese are the pioneers, that's where it all started back in the 1960s. But today we can say that wherever in the world business aquaculture, India with shrimp is a very good example also of this business aquaculture. It's technology driven, high energy and resources input. It is a successful new industry. Well, some of you in the room might say this is not always the case with shrimp farming, and I agree with you. Maybe we'll have some time to talk about later. But overall, if we look at the magnitude of aquaculture production today, we can definitely agree that it is a successful new industry. But the last word, and last but not least, all, I don't know of exceptions, all the business aquaculture is based on monoculture. Yeah. And why has that business aquaculture started only so late? Well, it had a lot to do with limited knowledge of the biology. Even still today, we don't know for all aquatic species how we can mature them in captivity, how we can set up hatchery production. So limitation in our knowledge. Then new technology that had to be developed, that became available, and that is in full development. I hear also about applications in India with artificial intelligence, with special counters, and you name it. And of course, the key factor that has driven the development of business aquaculture is profitability. I always use the same example. The Norwegians had selected salmon because they knew that this was a luxury fish and that that's a fish where they could make good profit. They never thought about to develop the aquaculture of herring, a very cheap fish even still today. And just again, I repeat, business aquaculture, monoculture approach. A crucial role in the whole development of business aquaculture has been the hatchery practices and as I put in the title here, the predictable availability, and maybe I should add an important keyword, not only predictable availability, but availability of high quality fry, fingerlings, post larvae, etc. Because I'm also one of the first to stand up and say, although we have been very much involved in hatchery developments, we have not always paid enough attention to quality. The focus was on the millions of post larvae that had to be produced, but in recent years we have seen that the quality of it's not because the post larvae survive, that they have a good physiological condition to do well. I'm not talking about mortality, but to do well once they are transferred to the ponds and might be exposed to stressful conditions. So still today, you might be surprised, but FAO still considers that hatchery activities is still the key to a successful aquaculture. And I think you all know this scheme where, okay, we go from the larvae to the market. Still today, I can give for each of these, I can still give examples, but the ultimate aim is that we are able to domesticate the animals. Once we can domesticate the animals, we can talk about selective breeding, and we are not dependent anymore on availability of be it the breeders or be it the fry from the wild, because we have had cases where the impact on the wild stocks was in fact leading under the suffering, under the pressure from aquaculture. And on top of that comes also the risks of introducing diseases. Still a very important slide and scheme, because maybe for salmon and tilapia we have very, very successful developments, but for many species, and I'm sure also a number of the fish species in your country, we are still relying on resources from the wild. So this hatchery activity is a very important industry when you see the value and when you see also the amount. I was surprised when FAO came up with the data that the hatchery industry is responsible for over 10 million tons of high-value aquatic species. Very, very brief, the different systems. We have the cage systems, for sure, also practiced in India, and we have them in very primitive structures, but over the years and on different continents, maybe it started in Norway, but we see it in the Mediterranean, we see it in China, in Japan, maybe also in coastal areas in India, where cage farming of marine fish, but also cage farming is practiced in freshwater in quite a number of lakes. It can be very sophisticated. Some of the units, for example, here in Norway, have very high automation and or producing very high quantities. It is sad that in many countries we see that these successful cage farming practices, well, didn't develop under good regulation, and I don't think that I have to predict that when you see this close concentration of cages producing marine fish and using trash fish as a food source, that diseases, harmful algal blooms, like you see here, that was in fact in Chile with the salmon cages that were put up too close to the coastline and caused a lot of eutrophication. So the cage farming, if done properly, no problem. You can be far offshore and have good dilution, but in many cases, and that's where I will in a moment come to that need for a turning point, in many cases the eutrophication is a very significant problem. The next, let's say the other important part of production systems are in ponds. And as you know, ponds, it can be freshwater, it can be brackish water, it can be marine. But when you see the pictures here, like taken in Indonesia with very intensive shrimp farming, using paddle wheels and very, very close concentrated areas that you can expect if a disease breaks out, that in no time you have a contamination over the whole area. At a certain point of time, I don't know if it is happening in India, but this is a picture in Vietnam, but you can see it also in Thailand. Then the big shrimp companies, they thought, maybe for better biosecurity, we have to go in a kind of greenhouse, but the big greenhouse systems, as you see here, but still applying that very, very high density monoculture flow-through system. And what happened? Today I'm already mentioning what you know, that the disease is vibriosis, but when it started, when it hit first China, but the main studies started when Thailand was hit in 2012, and where some of you will remember that everybody was talking about EMS, the early mortality syndrome, because nobody had an idea, is this virus, is this parasite, is this bacteria? But later on, and I will come to it in a moment, we all know today that it is caused by vibriosis. But before I go into these disease problems, let me finish with the other groups, long line production. So we had the cage systems, we had the pond systems, and then you have the long line systems, mollusk farming, I repeat, 17 million metric tons. An aquaculture where you do not have to bring feed, so the issue with eutrophication is not the case when we talk about the mollusks, and definitely not the case when we are talking about the seaweed that at this moment is very, very much, let's say, restricted to Asian countries. In the Western world, it's very, very minimal quantities of either the red algae, or the brown algae that are produced. Nonetheless, I repeat, it should be 33 here or 32, about 25% of the aquaculture production is made up of seaweed. So this brings me now to some new scientific developments, but that in fact have to be seen also into a historical context. And we have good data for the Mediterranean, and I'm using here the data for European sea bass and sea bream. But many of you in the room who have experience with hatchery production, be it of shrimp or prawn or fish, will most probably also make a similar reflection. What I show here are the accumulation of a lot of data for the Mediterranean industry. I have recently checked, because I was asked to give a similar presentation, I checked with the Invi company if the data from 2019 have changed a lot, and they say that they have improved, but definitely are still similar to the census they did in 2019 before COVID, with more than 200 hatcheries from all over the Mediterranean. And then you see, big surprise, 25, 28, maybe today 30, 35% average survival in both the sea bass and the sea bream industry. And this is not a small one, because we are talking about 1.2 billion fry produced per year. So this is a big industry. But what is the big surprise, and what is the big problem for these hatcheries? That they have a huge standard deviation. That they have some runs that very low survival, or even after 3 or 4 days that they can stop the operation. And then other runs that are very successful, 60, 70% survival, and unpredictable. And that has been the issue for quite some time, because look at data here that Professor Watstein published already in 1993, and okay, with Atlantic halibut, but very, very similar when you are working with other marine fish species. And you will understand why I'm using the word no antibiotics here, but just a regular run. And you see you have 2 hits and you have 2 misses. So already in 1993, it was hit and miss in the hatchery activity. And very important that already in 1993, he could demonstrate, well then you probably also know that there has been quite a period, especially at the turn of the century, that antibiotics were used, and we could indeed conclude this has something to do with bacteria, but not knowing, what kind of bacteria or what is the reason that when you are applying antibiotics, you have more predictable results. And you have more predictable results maybe for a few runs. At the moment that you develop antibiotic resistance, I don't have to tell you the story about the flexibility in the bacteria, the adaptability of the bacteria, and then even with the antibiotics, you have bad results. So when this happened here and people were talking about EMS, you see that we published some papers already in 2014, but we were very, very careful as a scientist. If you do not have the scientific proof, you have to use a question mark. But we were considering that maybe these sudden outbreaks, which later on, of course, were then linked with Vibrio harvey and later on even with other Vibrios, but that it was a microbial management issue, and that had to do with the practice of disinfecting ponds, the practice of disinfecting the water in the hatcheries that has been on for decades. But it was only in the period that the farmers, and no complaint to the farmers, but the farmers, they got so paranoid about the possible entrance of diseases in their ponds that they were using hundreds of kilos of calcium oxide, even setting up ozone treatment systems to kill all the bacteria in the pond systems. And as you will see in the next slides, that's where we could also then explain why it is called early mortality syndrome, how it comes that the bacteria are attacking early. So I was pleased to hear from Rina about the impression she has about the larvae meetings, but a very important one was the larvae 2017. And by the way, Rina, I'm happy to tell you that later today or tomorrow, the announcement of Larvae 2024, the live new Larvae meeting in September 2024 will be sent out today or tomorrow. But the 2017 one was an important one because there were many presentations dealing with microbes, microbial biology, microbial management, and it turned out that a lot had to do with the fact that suddenly, and I'm sure also some of you with experience in bacteriology will know that where in the past we were entirely dependent on plating techniques, and I'm not a bacteriologist, but the expert bacteriologists know very well that it is a very, very limited number of bacterial species that you can culture on plates. But when you can rely on DNA fingerprinting, when you can do a full genome sequencing as the prices were coming down, suddenly it became clear that in the aquatic environment, we are dealing with a huge diversity. I don't exaggerate by using the word huge. We are talking about thousands and thousands of different species of bacteria that occur in the aquatic environment. And I must say, I'm not good in all these emois and graphical presentation, but with the help of some of the students, we were able to maybe simplify the things, but try to explain that you are dealing with a lot of good bacteria, And for some of the good bacteria, we know what their role is, like a nitrosomonas, a nitrobacter, the role in the nitrogen cycle. But many of the beneficial bacteria, we still have not the slightest idea what they are doing. And I make a comparison. How often do you and me hear in the public media on the role that the bacteria are playing in our human body? And we are not just talking about the role they play in the digestive system, but some of the latest news we heard is that bacteria are even playing a role in the good functioning of the brain. So can you imagine what functions in the aquatic environment with such a good medium for bacteria that we are missing, that we do not understand? Of course, we have always been and should always be afraid and see how we can, for sure, remove the obligate pathogens. And then here, this is the opportunistic bacteria, where we had so little knowledge. What are they, and why do we call them opportunist bacteria? So in aquaculture, we have always had, of course, the good principle. Let's make sure that we do not introduce pathogenic bacteria in our system. And how can you do that? Well, a very good practice in biosecurity is you do disinfection. And if you follow the animation here, you will see, of course, when we do a disinfection, hypochlorite or calcium oxide liming or whatever, we kill the bacteria. But we kill all the bacteria. And you know, you put there a glass of water, sterile water, you leave it there for a few days, maybe you are adding a bit of sugar as a culture medium. Well, you come back in a few days and you will find bacteria in that glass of water. Because we will get immediately, well, immediately not within seconds, but within time, and that can be a matter of hours, we will have a recolonization. And the problem is that the recolonization is different when we are talking about the so-called R strategist versus the K strategist. This is a terminology that comes from ecology, be it in the terrestrial environment, be it in the aquatic environment. You have these colonizers, the quick colonizers, the R strategists, and then the more specialists, be it plants, be it herbs, whatever. So in the case of the bacteria that we are dealing with in the aquatic environment, the Vibrios belong to the R strategists. Some Vibrios also belong to the K strategists, but the majority of the non-Vibrios are K strategists, with the exception, of course, of the very pathogenic. And what are now these crucial differences? Crucial difference is that the R strategists, they are, well, as the word says, opportunists. They are very quick. They have a very high growth rate, but they require a high substrate per individual. So that means when they enter water where there are nutrients, and the nutrients that can be some of the feed, that can be some of the excreta of your animals, they are very quick in developing. Whereas the K strategists, and that's where all these good bacteria belong to, the growth rate is much behind the rate of growth of the R strategists. But this is the very interesting characteristic of the K strategists. They are very specialized and they only require a low substrate per individual. So that means when you reach a moment of competition, well, when there is competition, the K strategists will win, but not in the beginning when you are starting your culture. Then there is another phenomenon. He is now a professor but he was a former PhD student and postdoc at our lab, Tom de Voort, who has done a lot of work on quorum sensing, and particularly in the aquatic bacteria, because quorum sensing is a phenomenon a behavior phenomenon in bacteria that, let's say, the older among us never learned or heard about, and that is that what we call the primitive bacteria, we should rather reconsider the very specialized bacteria. They have a way to communicate, and they excrete signal molecules. Other bacteria or the bacteria of the same group can recognize, can capture, can in fact quantify the number of these incoming signal molecules. And when, and this is the explanation of the quorum, comes from the Latin quantity. So when quorum is reached, so when a bacterial population is reaching a certain density, only then, and that's remarkable, only then these bacteria will switch on the virulence genes and switch to a pathogenic status. This explains why for such a long time, and I remember it was Professor Karuna Sagar, if I pronounce it properly, who was also involved in these early studies on quorum sensing, and that it turned out that you do a diagnosis and find the presence of Vibrio harvii, and in some cases the Vibrio harvii, although present in the tank, in the hatchery in Thailand, for example, no problem, good survival. And you go to the tank just next to it, again, you detect that there is Vibrio harvii, and you get bioluminescence and high mortality. We didn't know at that time that it had to do with this phenomenon of quorum sensing. I do not have the time to go into all the details, because this is how science is evolving. So there are new phenomena, phenotype switching. It was an Indian PhD student at the lab who also did a lot of work on it. So it's a little bit even more complicated than just that quorum sensing in function of density. Certain conditions, so certain phenotypical conditions can also result in the switching on of that quorum sensing behavior. So here we see in animation what is normally happening in a tank. We have disinfected the water. Okay, we bring in the animals, we bring in the algae, we bring in some food. That means there are nutrients. And who is benefiting first? The opportunistic bacteria, the Vibrios, the R strategists. But if we have enough time, and this is just a matter of days, weeks maximum, the slow growers, the K strategists, will eventually also reach a high density. And once they reach a good density, they start to outcompete. And at the end, you will see that the opportunistic bacteria, they are still there, but they are not in high density. They do not express virulence. So this is also why we do not put a figure here on the axis. Why? Because that quorum sensing, that critical maximum density is related to quite a number of factors, like I was mentioning, how much substrate is available, maybe some other phenotypical characteristics. But it means that in some runs, the density that is reached here is below quorum sensing. Very good result. We worked in another tank where maybe we have just been adding a little bit too much algae or a little bit too much food, giving the opportunistic bacteria a chance to grow into a slightly higher density, reaching quorum sensing, bang, expressing virulence. So this explains very well the hit and miss in the hatcheries. And so typically, and that has been the practice of the last decades in the hatcheries in particular, more recent in the ponds, it is typically during a massive microbial growth that we are introducing our animals with that risk. If we take the time, and there are many, many good examples of green water technique and recirculation over a biofilter, as you will hear in a moment, that you are introducing your animals after this peak, at the moment that the good bacteria are dominating and in fact keeping control. This is so important about these good bacteria, not only their characteristics that we don't know yet very well in terms of maybe secreting enzymes, assisting in good digestion, producing immunostimulants. We still have to find out, but that they suppress, this is the important knowledge, that they suppress the opportunistic bacteria, particularly the Vibrios. So now it is very easy, and maybe some of the older among you will remember the Zoea syndrome and the bioluminescence, and in fact, problems in shrimp hatcheries. I have not been back in India in recent years, but I have been in Ecuador, in different countries in Asia in the last years before and the last 2 years after the COVID. And still today, I'm sorry to say, and I hope it is not the case, but there might still be a number of hatcheries, even in India, where they are still exposed to these problems because of lack of knowledge of that problem with the bacteria. So there are a lot of empirical observations which were very frustrating in the past, because we knew that it is going better working in that green water and not so good when we are working in clear water, but it is only today, with the knowledge we have, that we better understand. So the driving principle is, how can we reach a good balance between the R and the K strategies? And it is this balance that will determine what the risk is for bacterial disease outbreaks. And if you do not have good control over that balance, well, then you have unpredictable results. And this is what is still the case today in the marine fish hatcheries, for example, in the Mediterranean. So what are simple solutions, and what are the developments that I see happening in adopting now that new knowledge? Very, very simple. Use of protein skimmers. And I must say, I'm so impressed each time, just a couple of months ago again, in Thailand, in Malaysia, the protein skimmers that I see appearing now in the hatcheries, even in the land-based tank grow-out systems. Very, very simple. Okay, you can buy the very expensive equipment, but once you know the principle, once you know what that counter-current air bubble interaction with the water skims off the dissolved organic matter, and it is the dissolved organic matter that is the substrate for these R strategies. And maybe some of you will say, yes, but we are applying good filtration, sand filtration, and back filtration. Please realize you are removing particles, but you are not removing the dissolved organic matter. And that's where protein skimmers can really be, call it nearly magic. Then the other thing which we set up these simple recirculation systems more than 20 years ago, first in Thailand, then later in Vietnam. Rina will remember that in our laboratory already for the past 30 years, we are 40 kilometers from the sea, so we cannot bring every week the sea to the lab in Ghent, but we were in fact we were forced back already in the 1980s with macrobrachium, with the penade shrimp, to do recirculation. And now we understand why, with the good functioning biofilter, the one that you see here in front, you have the big tanks, either with macrobrachium larvae or fish larvae or shrimp larvae, and then over a protein skimmer, you bring it into different compartments with different substrates. And what is happening here? You get a very stable, mature microflora with a big diversity. Important to think about the big diversity. That means that you have to provide different substrates, not just the plastic bio balls, but crushed shells, small plastic material, coral, gravel, So this is what is being practiced, in fact, on an empirical basis already many years, but we now understand why they have such good results. And why? You have a long water retention time in the system, of course, and that's the purpose of the biofilter, remove the organic matter, but it gives the perfect opportunity to let the K strategists dominate over the R strategists. And that explains why these recirculation systems have such a good result. And I'm proud to show that based on the cooperation with our friends, in particular here in Thailand, what they do now, and I start to see it in Vietnam, I start to see it in Malaysia, hope to see it also in India, you continue with very intensive shrimp farming in ponds, but half of the area, and now you will be surprised, half of the area is used for recirculation. So it is no longer an inflow-outflow as it was in the past, so prior to EMS, water in and water out. No, after EMS, it is a recirculation system, and where you were only using 50% of the ponds for intensive 100 and more shrimp per square meter, but that you have the same amount of ponds where you put seaweed, tilapia, oysters, and you do a kind of recirculation. Or what CP company here is calling the zero exchange system. And you see, if you compare before EMS, then during EMS, and then after EMS, only half of the area is used for shrimp farming, and they have a much higher profit. I remember it was not easy to explain to farmers that using half of the area that you can have more production. The more production is mainly linked to predictability. And that's what the farmer does not always consider. You have to think over a full year, over 2 years, what is the chances that I will have run following run with good survival. And the picture you have seen before, microbial diversity and stability, you see the marine brackish water tilapia that they are using. In Thailand, they are using Caulerpa. In Vietnam, they are using Gracilaria. The Vibrio parahemolyticus, carrying the PIR-A and the PIR-B toxic genes, is still there, but not expressing virulence. So this is a very, very good proof that through quorum sensing, the critical bacteria might still be there, but if you create a suitable microbial environment, you will be able to prevent it. Very nice example here of land-based, and in fact, 80 kilometers from the sea, bringing in concentrated seawater and making seawater by dilution. 30 cubic meter, if I remember well, shrimp pond. Water is recirculated over the tilapia. From the tilapia, it is going to 2 tanks with Gracilaria, with oysters, and then finally trickling filter, submerged filter, and back into the system. So recirculation, land-based, now they have the commercial units, as you see here. This is the culture tank, and then here is a series of 6 tanks where tilapia, Gracilaria, mussels, sedimentation tank, and biofilter. So that brings me now to what you were expecting. Fed aquaculture, the aquaculture in ponds or in cages that without any you create an environment where there will be nutrient enrichment. You will try to feed just upon satiation, but you have some extra feed that is lost. You have the excretion products of the fish or the shrimp, so you do create eutrophication, whereas in the extractive aquaculture, remember we were talking about seaweed, we were talking about all the mollusks. This is nutrient recycling. This is in fact, as FAO is using that terminology, bioremediation. So I'm very, very much convinced that we need to see how we can integrate, how we could see some kind of interaction between the two. And we do not have to reinvent the wheel, because this is done already. These are pictures here from huge bays in China, some 200 kilometers east-southeast of Beijing, and where over very, very extended areas you see the seaweed production. So for a number of decades it was only seaweed, but then gradually you start to see the appearance of fish cages, and so the fish cages here are surrounded by the seaweed and mollusk farms. And indeed, this is big areas. This is a picture of Google Earth, 8 kilometers. So we are talking about an area here of, well, maybe close to 100 square kilometers where you have such integrated farming. This is the old cages. It's still an old picture that I'm using. I could show you the pictures where they are using the modern circular cages for the finfish, the yellow croaker. They have different species of fish they are growing there. But all surrounded, you see the white and the black boys. So some are carrying the long lines for the mollusks, others are carrying the long lines for the macroalgae. And you have, in fact, no time to go into the details, but you have a perfect integration and recirculation of the nutrients that, when you apply in monoculture, become a threat because they are a substrate for the R strategist. Here you don't give a chance to the R strategist because you have a continuous loop that is approach. So this question mark here, because we don't have the scientific culture, the monoculture approach that we apply today with the cages and with the ponds, that such a monoculture most probably creates conditions for microbial instability, and that when we might be able in the future to integrate, or if we can better study what the Chinese are already practicing for quite some time, but still should do some more scientific research, maybe polyculture is a way to create microbial stability. I'm finishing here to say that we have a chapter where all this concept is worked out in detail. It's an expensive book, but I can send a PDF copy of that particular chapter where you can find much more scientific explanation to what I have explained in this presentation. And I'm also referring to a paper that was published in Reviews in Aquaculture 2 years ago, where also in China, ecological intensification is one of the keywords that is considered. I finish with a slide here from FAO presentation. Microbial management, I think I have tried to convince you that in the future we need to go for more microbial management as part of the GAPs, the good aquaculture practices, but we need to admit, and this is the way I want to conclude also my presentation, our scientific knowledge is still limited. We have to admit that we need much, much more work on microbiome, understanding what the role of these bacteria is, and hopefully with better understanding we will be able to do better microbial applications in aquaculture. So where some of you might have seen, I don't know, Rina, if you were present at some of these global conferences on aquaculture of FAO, but where once I was invited to give a plenary talk, and I came up with the title Aquaculture, the Blue Biotechnology of the Future, but there was a question mark at the end of this sentence, because I was still claiming we have the possibility to go for blue biotechnology of the future, but we will have to do some scientific efforts. Today, I remove the question mark. I'm convinced we are on the right way. Aquaculture can and will become the blue biotechnology of the future. Thank you.
Mr. Sorgeloos for giving so useful, informative and wonderful lecture talk, and we are really are getting lots of idea in which direction we should do our future research also. And some of those things are really, I attended last week conference and last 2017 conference also, we talked about lots about the Quran sensing. So since then, that time I have started little bit to work in our laboratory also. You will be happy to know that not only the nitrosomonas, nitrobacter, we have also seen lots of archaea, they are also playing major role. And we are always thinking about the water quality, but we do not know their biochemical composition we are studying, but we are not studying the microbiology. We need lots of trained microbiologists in our country also. So thank you very much for this nice presentation. I am requesting you to send all these your papers PDF or even that list. I will circulate it among our participants and also among the students. Please send that PDF file if possible, and otherwise you can send me the list nowadays even from the internet we can, but sometimes it is not possible, they ask for some payment. So it is better you send me the PDF. I will distribute the soft copy among the participants and also among our students. So it is really nice. If you have some time, because I know that you have to go to some place, you have some time.
Yes, I still have 10 minutes. So
okay.
If there is time for questions, I'm available.
Before that, just I want to request all of us present here and also to you, we have to maintain 1 minute silence in the memory of Madam, and then we'll introduce ourselves. Just 1 minute silence, please.
Yeah, okay. Thank you very much.
It is really a special day for us. So okay, now I'm requesting our participants to just give their background so that you will get idea that with whom you have any question, any limited time. You have any question you want to ask? Anyone? Ask. If you have any question, you can ask.
Sir, it's really nice to hear from you, and then actually presently I am trying to do some microbial works. It's related with the biofloc technology. So recently I have set up 2 tanks, and then I'm trying to do those bacterial cultures, and then also it's a good technology, I think, but I think I will be requiring lots of help from you, and the many documents you share may be very helpful for us. So what in addition to biofloc, so to reduce ammonia, which types of technology we can add on those things? Like you talk about the protein skimming, so that will be helpful to make to reduce the amount of the ammonia in biofloc technology. So
yeah. Well, thank you very much. Very interesting question, and I'm happy that you bring up bioflocs. because we are talking two different systems here. And I fully agree, these are two systems that can resolve the problem of harmful bacteria and quorum sensing. But there is one big risk with bioflux. And before I start to further comment, let me first say I'm a big promoter, I'm a strong believer in bioflux. But, and I put but in capital letters, we need to know more about the bacteria that operate in the bioflux. And what I have seen is that in many cases, also some of our students, some of the applications in Indonesia, many other places, you can have very successful runs with bioflux. You can have many successful runs with bioflux. But you can sometimes have a disaster with your bioflux. Because if you do not maintain the right composition of the bacteria, what will happen is that by increasing the CN ratio as you need to do to stimulate the growth of your bioflux, you stimulate then the bacteria that will be the pathogens, opportunistic pathogens. So I repeat, strong believer in bioflux, but there is still a risk involved because we do not have today, we do not have the tools to know about the right composition. We do not have the tools to know, oh, things are going wrong, we have to stop adding nutrients for the bioflux. So what I'm highly recommending is, please continue to search and research on bioflux, but I'm a little bit skeptical when I see what has happened in Vietnam and a number of other places, that we tell the farmers, this is the solution. Dump in the molasses, apply it at big scale, wow, very good success, oh, we are very happy. And then 2 months later, 3 months later, a disaster. Yeah. So positive when it comes to the bioflux, but we need more research. Yeah. Then the other system where you were talking about the protein skimmer, and particularly the biofilter. You cannot imagine what a big promoter I have become of simple recirculation systems, not the sophisticated ones that you buy in some of these, I will not mention the countries in Asia and in Europe. No, look, and I will make sure that I send also that article together with the chapter to Rina. Yeah. Simple, really simple biological filters with locally available materials. You don't have to buy expensive bio bowls or other products. And you create, and okay, when I say now that this is magic, to some extent it is magic, but you create a very stable, mature microbial community. Yeah. We do not yet fully understand what that microbial community consists of. But I can bring you to places, and it must be the same in Thailand. And I like so much now when I'm traveling to, the last one was in Malaysia, that I say, bring me to old farmers. And I visited farmers that have been 30, 40 years in production, not huge scale, but still a significant scale, using biofilters, using recirculation systems, with plants, with all kinds of materials. And these farmers, they say, well, we learned it from our father. We are applying this already for different generations. Many people are laughing with us that this is the old-fashioned way. But with the knowledge that we have today, this is where we need to study what is the composition in the different compartments, with the gravel, with the crushed shells, because we will probably see, that's what the microbiologists are telling me, some of the bacteria prefer a carbonate substrate, others, it might be any surface area, the coral, the broken coral, okay, it will contribute also to the alkalinity in the system. So the biofilter is magic, but it works. And it works for years. This is not something like in the bioflux, where once in a while, if you were lucky, it could be a year without a problem. If you were unlucky, it could already be after a couple of months.
So, Professor Sorgeloos, I just want to mention, just add something regarding the recirculating system. I learned from your institute, and then since 1998 till date, we are using recirculating system, and I am using the locally available materials because I didn't have money to purchase bioballs, because at that time the cost of each bioballs was Indian rupees 10 rupees. I did not have that much of money, so I have used the pebbles and broken earthen pot. Those pots are used for the culture of the plants in our country, and after their break, people are throwing them. I brought them, I cut them into small pieces because they are serving as very good substrate for the growth of the microorganisms. And you see, in summer months also, because these are earthen pots, and the temperature, they are also maintaining the temperature in the culture system, and along with that we are growing different plants. It is a combination of this microbial population as well as the plants, because nowadays we want more crops. Some crops, we are growing plants like azolla, lemna, spirodella, all these are growing, and we are using them for the production of fish food, because we need the alternative source of protein, and they are containing a huge quantity of proteins and other things. So if we use the biological filter of these microorganisms along with that some plant, and we'll get more production. And you told that instead of monoculture, if we go for the mixed culture in the recirculating system, in different compartments, if we can maintain fin fishes and so also mollusks and also these prawns, so if we can put them, and if we go for the monoculture, then we can, because some of those these animals, they are producing some pheromones or some other chemical substances that may create control the other population. So I think recirculating system, though it is our age-old practice, also the composite culture or integrated farming, these are really very good practice, and we should follow this. We should not remove them from our system.
Sir, the recirculating system. Okay. I have been working with ma'am, and recirculating system is very good, and I have been doing that. But in the bioflux system, the difference is in the production. There is ability of producing huge amount of the fishes, but when we go into with the mechanical system, but in India when we try it costing 5-6 lakhs, and even sometimes 45 lakhs if you want to do with a very huge system. But the thing is that in bioflux system we reduce the biofilter system. You keep the fishes as well as the bacteria in one place. So, and moreover, but there is requirement of regulation of water quality. It's very important, like ammonia and all. So, and now, and one more thing is that now you are talking about the bacteria. In India, we have many companies they are preparing the probiotics which are species specific. For example, you want to reduce the ammonia, they are providing you those specific bacteria. For example, simultaneously, there is an increase of ammonia. So you can buy a bacteria and then put it in that tank, and within few 7-8 hours, it's the problem is solving. So that's I have been doing, but I think it depends. Yeah, if somebody can fail if they don't know how to maintain the bacteria, that is the thing. First we have to know the maintenance of bacteria, then after that fish will survive. Thank you, sir.
Anyone want to ask question? Anyone?
Yes,
Jitu?
Sir, so wonderful talk, Professor Patrick. And I just want to ask how these biofilters only select the K-selected, what is the mechanism? How they are preferring only the K-selected bacteria? Biofilters.
Well, I'm not sure that I understood the question well, but the functioning of a biofilter takes time. And this is also something which is important to know. It can take 2-3 weeks or even longer before you develop a mature microbial community that is very diversified. And I'm sorry that I'm using the word magic. A scientist should never use the word magic. But there are still so many things that we need to realize that we don't understand yet. But we know from experience, and not experience of the last years with the knowledge we have, but when we listen to the old farmers in many, many parts of the world, that type of developing a mature biofilter that takes weeks, and then is really, really stable, because then what we do in the lab, and what maybe you also do in the lab, you are adding ammonium chloride, so before you switch it on to the fish, and you check the next day what your nitrate, ammonium levels are, so that you can prove, okay, this microbial community, I'm sorry, which is magic, huh? We don't know how many hundreds or thousands of species, but this is where both for the bioflocks, both for the biofilters, we need much more research on what is the composition, what is the composition in this compartment of the biofilter, what is the composition in the other compartment, and then not only that in terms of composition, but what are the functionalities. And I think we will be surprised in the future to see what important role the bacteria are playing in digestion, immune stimulant, a lot of physiological parameters that we are not aware of yet. Of course, you are scientists. This is a discussion where we can understand each other, that we still need to learn a lot. But when you tell this to a farmer wants a solution yesterday, not tomorrow. He wants a solution yesterday.
Thank you, Professor.
You want to ask something? Quickly.
Just a basic question. Like
Come. Everything is important.
Hello, sir. Just for the R strategist, like, do we have the Vibrio only species, or there are other infections? Like Vibrio is...
Can somebody closer to the microphone repeat the question?
Sir, I wanted to ask, like, is the Vibrio is the only known R strategist in the microbial consortium, or there are other species, Shigella, for example, and like other species which are there for the R strategist? I just wanted to...
Yes, it's not just Vibrio. Again, I will not say that I'm simplifying things, but it is indeed much more complicated, because this is where we have already the very first problem. Some of the Vibrios are R strategists, and some Vibrios are K strategists. Boom, that makes it really difficult. So the Juvenella you mentioned, there are quite a number of other species that either belong to the K or to the R strategists, and where we need more knowledge. I'm sorry to say the role of bacterial research linked with your specialties on nutrition, on immunology, need to go hand in hand. This is where I also know your laboratory is very famous for the nutrient, all the papers on diets and diet ingredients. Please realize, and I think it is one of the next maybe days, that Sashi Koshik will also give a presentation. Well, challenge him about the role of bacteria. But I remember years ago, but now Sashi also agrees with me, that running experiments where you are testing in treatments a particular diet A, B, and C, the colonization of the bacteria in treatment A, treatment B, and treatment C can be different. Why? because the substrate in A, B, and C is different. Bacterial selection A, B, C might be different, and you might have a response that is more influenced by the bacteria than by the diet. So very, very important also the role of bacteria in the whole system. And in many cases, again, this is not a very scientific word, but we are working in a black box.
Yes, it is really very interesting to know the role of microbes in our diet also. Yes, it is correct. You are 100% correct.
Well, this is why some of you might know. This is why we developed, and many students, Indian students as well, have done their PhD in the axenic artemia system. So this was the first time. You cannot do that with shrimp. It's much too complicated. But we have been able to prove in a bacteria-free artemia system that the role of the bacteria in the nutrition very crucial. Very, very, very crucial.
I think it is all of them are very much impressed with your lecture, and they are getting lots of idea for their future research also. Me too also, and I'm really eagerly waiting for the next larvae. You have told me very pleasant thing to me. I mean, this year I have missed that one. You have organized online. So last time it was online. So I'm not enjoying that much. So now I'm waiting for the offline our larvae. Definitely I'll come to Ghent. Thank you very much. Thank you.
Yeah, it was a pleasure to. Well, it's a long distance, but still I can see all the people around the table. I wish you good luck in your research. Feel free if there are. Exchanges through email where we can share ideas or even share how limited our knowledge is and admit what still need to be done. Thank you very much, and I hope in the future there might be an opportunity to be back live at your university and meet with you.
We want that you should come physically in our university, and we are waiting for that day also. Thank you, Professor, very much.
Okay, bye-bye. Have a good day.
Bye.
Bye-bye. Bye-bye.